The next prize winner is Antigone Martinez, and she won the Maltz Prize. You can come on up here, Antigone. And the Maltz Prize winner is named by the Lieber Prize winner. So this is a namee of Dan's. Antigone, her position is as a research scientist at the Nathan Klein Institute, and she's an associate professor of psychiatry at Columbia. Antigone is a cognitive neuroscience scientist, and she's studying brain mechanisms of visual attention and social cognition as they are impaired in schizophrenia. Now, I have to say, when I was thinking of her contributions to science, amidst today's speakers who are all absolute luminaries in the field, more at the end of their career than at the beginning. So I've named Antigone a developing luminary in our field. And what's interesting is that people who are developing luminaries and younger people have more tools at their disposal. She's using the tools of modern human neuroscientists like fMRI, imaging, and EEG, and brain stimulation paradigms like TDCS to identify and to treat the brain circuits that are involved in social cognition. She's already an outstanding developing luminary. Antigone.
Thank you. Thanks, Carol. Thank you for that luminary, I don't know, label that will carry with me now. And thank you, obviously, BBRF, for this award, and the Maltz family means a lot. It's very significant to me. I want to start first. Dan really, let me get this, make sure this works. No, this is the one that's not working then. I'm just going to put it up here so that other speakers don't use it. There we go. All right. Dan did a really nice job, actually, of setting up this talk for me. He spoke about early processing deficits in the auditory system and how these can cascade forward and contribute to higher order cognitive impairments. So I'm going to kind of follow along on that same theme, but I'm going to take you to the back of the brain into the visual system and talk to you about some of the recent work that I've been involved with. Okay, whatever. It's fine. It's okay. I can live with this. All right. So first of all, I just want to to get us grounded. We'll focus on the left side of the screen. This is just on how visual information is typically organized in the brain. It all starts in those red V areas in the back of the head. That's the visual cortex. From there are three main visual pathways. One feeds ventrally. It's not there. I don't know why. It's weird. Okay, there we go. There it is. One pathway feeds down the ventral portion of the brain. That's called the what pathway. That pathway is important for things like object recognition, face recognition, identifying objects. The other pathway, these pathways, by the way, are distinguished by their functional properties. The other pathway, one that I've worked on for a long time, Dan's done a lot of work on also, but I won't be talking about today, is the dorsal stream, the dorsal pathway that sometimes also is called the where or the vision for action pathway. And that's really important for spatially localizing objects in space. Things like, well, Dan showed that tracking objects in space, the soccer paradigm that Dan showed, where you have to follow a target with your eyes, things of that nature. But in recent years, really very recent years, maybe 5 years, a third visual pathway, very creatively named the third visual pathway, has been getting a lot of attention because this pathway, unlike the other two, is in humans uniquely specialized for social perception. And, oh, God, there we go. All right. This pathway is specialized for social perception, by which I mean the ability to use cues that are in visual information, to use cues social cues and visual information to extract meaning out of it. So meaning that is socially relevant, things like understanding someone's facial emotion, facial expression, their body gestures, their language. This pathway, this third visual pathway, is specialized for those types of processes. And like the other two pathways, it starts in the back of the head, it starts in primary visual cortex, that's the little yellow V1 over there, and then it feeds forward. It passes through first this another very creatively named area, the middle temporal area, MT, which lies in the middle temporal cortex. This is an area that is, it's like the hub, the motion processing hub of the brain. It is the center of motion processing. It responds very strongly and encodes simple motion, like the speed that something is moving in or the direction it's moving in. From MT, the visual information flows now into the superior temporal sulcus. Those are those blue STS regions. And these areas really are the part of this pathway that are important for social cognition. These areas respond to face emotions, but they respond especially strong if that face is in motion. So this is the STS regions receive those social cues. The takeaway point here is that the third visual pathway relies on motion. So social cognition relies on motion. So MT is part of this pathway because in order to understand social interactions, we need to understand and see and perceive the motion around us, right? So we use two paradigms to tap into these different types of motion processing, the simple motion in MT and the more social motion in STS. One is just a simple moving dots task that gives us a measure of motion sensitivity. I don't know if you can play that. It's a really simple task. You just have to look at the dots and you just say, are the dots moving left or right? The other task is a little bit more higher order, but also very simple, if you can play that. This task is just actors using motion, right, to display an emotion. And again, really simple. There's 4 or 5 different motions, and the participant identifies the emotion. And in both cases, with both of these tasks, people with schizophrenia perform worse than matched controls. And actually, not just people with diagnosed schizophrenia, but people that are at risk of schizophrenia, and even their first-degree relatives don't do well on these tasks. So, oh God, okay, I'm missing, oh man. All right, we'll just leave it at that. All right, so what these behavioral patterns point to is they point to this link, right, between motion processing and emotion processing. But to really understand what's happening at the neural level, we use fMRI, and we look to see what's happening in the brain. So we administer both of the paradigms, the motion task, the emotion task. We administer it to control subjects on the left, and then also to a group of people with schizophrenia. And hopefully what should jump out at you is that the dot task activates MT, as you would expect, and it does so very strongly in controls, not so much in patients. And similarly, the face task activates that STS, those STS social regions, and again, not so much, so very much reduced in patients. But the key here is that the degree of activation or disactivation in these areas predicts behavior. So people that activate MT strongly perform better behaviorally on the dot task. And same people that activate the STS strongly, they are better at recognizing faces. So we see that there's this link between the behavior that we're measuring with our paradigms and the underlying neural activity. So the question then, so now that we've shown that the patients with schizophrenia have this disruption, right? Their neural activity is reduced along that third visual pathway. The question then that we asked was can we use some new techniques to restore that function, to improve that function? So we are starting, or just finishing the first of hopefully many studies using a technique called transcranial direct current stimulation. What that is, TDCS, it sounds a lot fancier and scarier, really, than it is. It involved applying a couple, or electrodes, small electrodes on the scalp, delivering very weak current through it. The TDCS device is basically, it's like a 9-volt battery, and it delivers current, but the current is so weak and so dispersed, like diffuse on the scalp, that it's painless, and people just report feeling like tingling underneath the electrodes. So it's safe, it's painless. It's been used in other studies. There's numerous studies that have used this technique to show that you can improve, or they claim that you can improve attention and decision-making and other sort of cognitive processes. So our goal was to use this to see if we could improve face emotion recognition, because, and like Dan mentioned before, and I don't think that I highlighted this at the beginning, but the reason we care about face emotion recognition is that face emotion recognition is a core component of social cognition, right? And as Dan mentioned, social cognition is the #1 thing that predicts how well a patient will do long-term and in life. Like how well we're likely to have a job and social relations. So social cognition is an important thing. And face emotion recognition, this simple little task, really taps into that process, or that cognitive process. So that's why I care about this, and that's why we're using TDCS to see ultimately, can we improve that? So the goal was, let's apply the TDCS current to that motion area, to that MT area, and then we hypothesized that we're gonna look downstream and see if we improve this motion area, would it also improve that downstream social cognition areas in the STS. That was the goal, that was the simple sort of design. So the way we set about doing this was to use, and this is important, a personalized approach. So we first took, these are three sample subjects. These are all patients with schizophrenia. We took them, we put them in a magnet, we localize MT. MT is really easy to localize. It makes it a really easy and really good target for TDCS, because you can find it in individual brains. Unlike STS, this one it's very simple to localize. So we decided that's why we picked it as a target. We put them in the magnet, we find their MT area. We use those maps of where their MT area is to make a model of where we should place the TDCS electrodes. So every subject gets a different type of placement on the scalp. The electrodes are different. The amount of current that goes through the electrodes, the polarity of that current, it's all personalized. So you can't use like this one-size-fits-all montage, because as you can see, like everyone is a little bit different. The way that you need to really get to MT, because we're trying to focus the current to optimize the flow of the current through MT, it's a little bit different in each person. So this part I wanted to emphasize, because this personalized approach actually turns out to be key and very, very important. All right, so that's it. So we do that, we decide where the electrodes are, we do that, we administer the task. We administered it simultaneous with EEG and also with FMR. The subject is wearing, like 64 electrodes. We can stimulate, it's very fancy. We can stimulate from any one of those electrodes. Simultaneously, we can record the brain activity, the underlying brain waves from the other electrodes. And if that weren't enough, we also do the same thing. We put them inside the MRI magnet with a MRI safe cap, obviously, and we do the same experiment. So I'm not gonna have time to show you the EEG data, but believe me when I tell you that it aligns really well with the imaging data and the behavioral data that I'm gonna show you now, which is not that. Which is also not that. Okay, which is this. And I'll just go through this very quickly. It should be, hopefully it doesn't need me to explain much, but what you're looking at, the two paradigms, the motion dot paradigm on the left, the face paradigm on the right. On the top, let's start with the top left. The dot paradigm, when we use the sham condition, I don't know if I mentioned that we do this experiment with the comparison is always an active. So we're actively stimulating MT, that's an active TDCS, and we contrast that, or we compare that to a sham condition, which is basically with TDCS is off, right? So that's how we get a measure of whether or not TDCS is working. When the TDCS is off, like we saw before and as expected, there's not a lot of activity in MT, but when you turn the TDCS on the bottom, then MT suddenly activates, showing that we are restoring function in that MT region by stimulating MT. But that's our target. So this was really cool, but maybe not as surprising because that is we've targeted MT. But what's really cool is that if you look, again, we're still stimulating MT, you look upstream, we can also improve function in STS during the face of motion task. Thank you. All right, so let me just, one last point that I wanted to make, if I can get to it, I don't know, I'll just say it out loud. To the degree to which people improved with TDCS, TDCS doesn't only improve, it improved the activation in these motion and emotion areas, but that improvement went with better performance behaviorally. So the more they activated, the more they engaged these regions, the better they were at performing the task. So I'm going to just wrap up real quick here and tell you sort of where we're going with this, because so far I've just shown you these very lab-controlled paradigms. Now we're looking at doing similar things, but we're using movie clips, and we're looking to see whether we can change the connectivity between brain areas by stimulating MT. And I'll just cut to the chase and tell you that yes, we can. This is preliminary work, but this is kind of cool that the MT, the connection, that motion to social pathway is improved, is strengthened by applying this TDCS to the MT region. And, okay, and that's it. Look, I'm almost done. And I'll just to sum up and sort of reiterate some of what Dan also said, that deficits in social cognition, they are critical. They're bottleneck to functional recovery. That's why we target them. Some of the data that I've shown you now hopefully shows you that these methods, these brain stimulation methods, may actually prove to be important for restoring some of these functions, and in this case in particular social dysfunction. And where we're going with this is, God, you know what it is? It's not that it doesn't work, it's too sensitive, just so you know. Yeah, there. Where we're going with this is to take some of these lab-based findings and into more like personalized interventions and pairing them with repeated TDCS administrations, not just that single session, and maybe even developing, actually this already exists, little caps that you can take home and do have the stimulation delivered remotely at the comfort of your own home. So that would be cool, and the goal, of course, long-term is just to improve function and improve the lives of patients. So with that, if I can get to my thank you slide, there we go. I have a bunch of people to thank, and some of them are here, and I'll take questions.
Thank you, Dr. Martinez. Questions from the audience? Well, I'll start it off. From what Dan told us about what's wrong in schizophrenia, you told us about a very specific treatment. You would imagine, would you, that you have to, for any single person, use 4 or 5 of these specific treatments?
Yes, yeah, and so that's where we're going with this, actually, and that's a good point. And that's what, like, TMS treatments for depression work that way, that the protocol is that people come in, they get their TMS every day for 3 weeks, and that seems to really improve depression. So the idea is that with TDCS to really, because what TDCS is doing is increasing the connection between areas, to make that connection, the strength of that connection more long-lasting. Yes. We're actually we just started a project to do that. We're having people come in twice a day for over a week, and then testing them before and after.
You're putting this into the therapeutic area right away.
Yep, we just started a project to do that. Mm-hmm.
Any questions? Any more questions from the audience? One, yes. Wait, can we have a microphone?
How long do the effects last after
See,
treatment?
Jeff, you had to ask that, right?
I can ask a different question.
Yes. So we've only tested out About less than an hour. And I suspect, I can't tell you how long they last. I suspect it's not that long. And that in order to get really long-lasting changes in plasticity, you need these repeated sessions. And maybe pairing those sessions with cognitive remediation, for example, which is another thing that we're exploring. But yeah, no, you're right. These effects are not, with one session, are not that long-lasting.
I think the field is starting to explore other kinds of stimulation, more potent and long-lasting, like deep brain stimulation.
Yes.
Stimulations that are used in Parkinson's disease, epilepsy, we're now importing into schizophrenia.
Yes. But this is very much less invasive, and so
Absolutely.
we're hoping that it has a future.
With that, we'll say thank you to Dr. Martinez, and hope we see her here in another 15 years.
So thank you.